today. This is chapter 6 in your book. And so what is genetics? Genetics is the study of inheritance or expression of inherited traits. So there are some syndromes that are associated with genetic problems. What a syndrome actually is, if we talk about a syndrome versus a disease, when they call something a syndrome, it has to have a certain group of signs and symptoms that occur together at the same time in order to be deemed that particular syndrome. So these are just some vocab words that you're going to need to know from this chapter. Phenotype is what I actually exhibit when you look at me. So that's my physical, my biochemical, my physiological traits. When you look at someone, I have brown eyes, I have curly hair, I have straight hair, whatever that may be, that is my phenotype. These can occur as a result of genetic factors, something that's been passed down to you genetically, or maybe possibly through environmental influences. Genes are the hereditary units that are transmitted from one generation to another. And okay, so chromosomes, there's the double helix. You see the twisted ladder to the right. That is our DNA in between that make the rungs of that ladder. Chromosomes are found in the nucleus of a cell. And if we're talking about a somatic cell, which is every cell in our body besides our sex cells, the somatic cells contain 46 chromosomes. We get 23 of those chromosomes from mom and 23 of them from dad. And those chromosomes contain the DNA that will decide what we look like and everything about us. So, and DNA can duplicate itself. I'm sorry. There we go. So normal cell division. Now this is dealing with somatic cell division. We talked about this in histology. Mitosis is that somatic cell division with that those phases that we discussed. PMAT. P is for prophase. M is metaphase, anaphase, and telophase. The function of mitosis is just to create an exact copy of each chromosome, and it will distribute an identical set of chromosomes to each daughter cell. And in your book, this talks about, it's on page 188, the mitotic cycle. And the S phase is probably one of the most important phases that you might see asked about, because that's when DNA replication is occurring. So it starts out with mitotic division, then it goes to G1, and then into S, or replication phase, where that DNA is replicating itself, and then G2, and then we go back into the mitotic division again. This whole phase, like G1, S, and G2, are all kind of interphase that was always referred to as the resting phase or the resting cycle of this. But that is what's occurring during interphase, and then we start mitotic division again with prophase, metaphase, anaphase, telophase, and then it goes through interphase again, which makes up this G1, S, G2. On page 189, it's kind of showing you what a chromosome looks like and how we've got a short arm and a long arm, and it's gathered in the middle. I always think of like a belt gathering your dress in the middle. And that middle part is called the centromere. And if you look to the left here, the entire chromosome here, double chromosome, is a chromatid. So again, the stages of mitosis, during prophase, those chromosomes are going to be lining up towards metaphase or towards the midline. I always think midline with metaphase. Metaphase are where they are lined up kind of at that equatorial plane in the middle of the cell. The long and short arms are joined again at that centromere or that cinched waist, and each identical half of that is called a chromatid. So then during anaphase, the third phase of mitosis, the chromatids are in the process of splitting. So that double chromosome that has the gathered waist now is going to split vertically down the middle into two different chromatids. And in telophase, then cytokinesis can occur, which is the actual formation of those two daughters, where it's actually split apart. Now, sex cell division is meiosis. This is a two-step cellular division of primitive germ cells. So it's either oogonia, which is the egg, or spermatozoa, which is the sperm cell. And then we will develop from this mature germ cells. And those mature germ cells only have 23 chromosomes each. So mom has 23, dad has 23. Meiosis will reduce the number of chromosomes by half, and then they become those germ cells. So remember, we start out with 46, and then they're split in half, and those become the mature germ cells that only have 23 chromosomes each. A primitive germ cells are diploid, that's before they've divided, so they have two sets of 46 chromosomes. Mature germ cells are haploid, so they only contain the 23, or a single set of chromosomes. The first meiosis, because we go through two meiosis cycles. During the first meiosis, members of each pair of those chromosomes will line up and exchange segments at their contacts. If you look at these little guys, it looks like they're kind of wrapping their legs around each other. So you've got one set of chromosomes and they buddy up next to another set of chromosome, and they kind of wrap their legs around one another and exchange those segments. That little area, or that exchange, that segment, those contacts are known as chiasmata, and that's just where those segments switch. During this first meiosis is when a condition called non-disjunction can occur. Occasionally what will happen is both chromosomes that we're crossing over don't separate, and so both of them go to the same cell. So that means we've got an extra chromosome. This can occur during first or second meiosis. In Down syndrome, or trisomy 21 is an example of this. So tri means 3. So rather than only having two chromosome number 21s, we have 3 because they did not separate. And that's called non-disjunction. Now, during second meiosis, each chromosome splits longitudinally or vertically. No replication of DNA occurs before the second meiosis. And after this splitting, the two cells are formed with the right amount of DNA at this point. So non-disjunction of second meiosis is when a chromosome does not split. The one daughter cell has a full chromosome and the other one has none, so they don't split. The Lion hypothesis is a hypothesis that was formed, I believe this woman's name was Mary Lyon, and what she found was that sex chromosomes are designated XX in women, obviously, and XY in men. And during early embryonic development, the genetic activity of one of those X chromosomes in each cell of a female embryo is inactivated. So her belief was that we have two X's, we start off with two X's, and then one of them kind of becomes inactivated. And the inactivated chromosome forms a structure known as a bar body. And if you look at figure 6-7 on page 191, it shows you what that bar body is. And if you look at it on a slide, it's just kind of a dark oval. And it was her belief that this was just an inactivated X chromosome. And this is what that looked like. So it's kind of in the nucleus. You can see the cytoplasm encircling the nucleus, and then that little blue area on the picture down in the left corner is supposed to be signifying what that bar body might look like. Okay, deoxyribonucleic acid is DNA. It carries all of the genetic information. A nucleotide is the basic unit of DNA. So it is a nitrogen-containing base. It has a 5-carbon sugar that is deoxyribose in DNA. And then there's a phosphate. The 4 bases that we see in DNA are adenine, guanine, thymine, and cytosine. Adenine will bind to thiamine and guanine will bind to cytosine. And the ratio of those is always equal. So the ratio of adenine to thiamine is equal, as is guanine to cytosine. They will come together and match up. So if you have an adenine on one side, it's going to match up with the thiamine on the other side. And they're going to form this double helix or this twisted ladder. And if we have a sequence of 3 bases, that's called a codon. Now we're going to talk about ribonucleic acid, RNA. So what is the difference? RNA is ribose rather than deoxyribose. It is only a single strand, and anytime you see ribose, think protein, because that's what it's in charge of, is producing amino acids, which are the building blocks of protein. So the genetic code of DNA is transcribed into RNA, and that is so that RNA can help produce amino acids, polypeptides, and protein. In an RNA single strand, the base uracil now replaces thiamine that we saw in DNA. There are 4 types of RNA. There's messenger RNA, which is the blueprint for coding of proteins. There's transfer RNA, which transfers those amino acids from the cytoplasm of the cell to that messenger RNA so that it can go help make protein. There's ribosomal RNA, which combines with several polypeptides to form ribosomes, which are again a protein. And then heterogeneous nuclear RNA is just a precursor to that messenger RNA, and it's going to be found both in the nucleus as well as the cytoplasm of the cell. So anytime you see RNA or ribosomes or anything like that, think protein, because that's what their job is. And if you look at this little flow chart on page 192, this is a production of protein from DNA. So we start with DNA, and that DNA is transcribed to messenger RNA, and that messenger RNA will take it to the ribosome and then create protein. And then you can see also transfer RNA can have a role in this too, and transfer RNA brings those amino acids to the ribosome to make proteins. I'm not sure why my computer's spinning, but we're going to hope that it keeps on working. And it's not. All right, I'm going to stop this for just a second here.